Okay. You all can hear me, right? All right. Thanks for sticking in all the way to the end. I certainly appreciate it. And it's my pleasure now to cap us off with what I call the algorithm for precision medicine. I hope we'll tie together a lot of the things that we've seen today. Of course, I have to begin with a fairly important disclaimer. I have no medical training whatsoever. So if anything in this talk resembles medical advice, do not take it. Or at least consult your physician. Or Tyler, please, consult somebody with any kind of actual biological expertise. And by the way, there's a lot in this talk that does resemble medical advice. And that's because prior to 2017, I did programming languages. In fact, I put together a little graphic of like where I actually used to do my work. And it turns out that like this is my home. Like ICFP is where I published most of my stuff once upon a time. And if you rewind back to like 6 years ago, this is the balance of my publications between PL and medicine. So it's a little different now. I was kind of sad almost when I saw this morning when I checked it. I looked at Google Scholar and it turns out my top publication is actually now in medicine. It knocked off abstracting abstract machines. And it's of all things, it's a paper on using metformin to save diabetics with COVID, which, hey, it's great. Saving lives is great. But gosh, I do miss type theory sometimes. So I'm not alone, though. Turns out there's another computer scientist whose top publication is also in biology. It's Alan Turing. Alan Turing's top paper is in biology. That's because he was using computational principles to model things like morphogenesis, which turns out to be the right way to model it. So computation and biology go way back. Second disclaimer, not only is there medical advice in this talk, all of it is computer generated. So just more reasons not to follow it. In any case, I am going to make some claims. So one of my claims is that when we look back at the history of medicine for this century, we will see that the greatest drug in terms of intervening and making a difference in human health was actually data. And I firmly believe this will be the case. I also believe that as a field, precision medicine is in some sense what delivers data as a drug. That is what we're doing. Henceforth, precision medicine requires computation. We have no choice but to invoke it. And the question I have for everybody here is the algorithm for precision medicine, is it inherently or most naturally expressed in a declarative way? I think the answer is yes. So I'm going to show you, not formally, because if it was done formally, I would have answered the question, what the algorithm looks like with lots of examples, some high-level structure, and my attempt to render fragments of it in a declarative way. And it just seems like the most natural way to express it. And the algorithm for precision medicine, by the way, is what I'm referring to as the very high-level process of going from a patient and their information or their data over to a set of recommended treatments that are truly tailored to that patient. I do think that it is most naturally declarative. So there's three components to the talk today. The first is, what is precision medicine? If your background is primarily on the computational side, this could be a new term to you. I want to give you a sense of what it looks like, secondly, and I'll just do that by showing you examples of patients that we've actually helped at the Precision Medicine Institute over the past 6 years. And then I'll actually try to distill the algorithm from all those specific examples. And in the course of doing this, you'll end up figuring out why I jumped from PL over to academic medicine. So, what is precision medicine? If you go back in time to 2011, the first place it appeared was actually this report from the National Academies. And in the back, there's this glossary of terms, and they try to define this term they've just defined, and it's like this essay-length debate and argument with itself over what it is. But what they came up with is that it's really all about tailoring treatment to the individual characteristics of patients. So that's a fair definition, and that was enough for President Obama to launch the Precision Medicine Initiative in 2015, and then everybody decided that they were doing precision medicine. So all these people said, this is what precision medicine really is, and all of these things are, of course, they're a part of precision medicine, and one of the jobs I had at one point was to wrangle all these definitions back into a unifying definition. And the one I came up with is that precision medicine is really about optimizing health with data. That seems to encapsulate both the original definition from national academies plus everything that everybody else was claiming was precision medicine thereafter. So since it's optimization, I do think of it as a process, an algorithm, if you will. And the input here is patient data, and the output is a set of potential treatments for that patient. And I would even go as far as say it's not just a set of potential treatments. We also want the rationale for the treatments too. We don't want just a black box, just try this. And we know this from experience, that doesn't work in medicine. Like if you tell a clinician, just try this thing, the black box said it'll work, they will not use it. They will not touch it. And so those explanations really do matter. But at a high level, the process looks something like this. You've got a patient, and you begin to pull out data. And the piece of data that really got everybody excited about precision medicine was the sudden clinical accessibility of the human genome. Like the fact that you could sequence a person's genome and get access to it, and a clinician could see it. I mean, that was groundbreaking. But it also meant that you couldn't avoid computation at this point. I mean, genomes are massive. To construct them, it's massive amounts of data. To distill them down, it's massive amounts of data. To interpret them, it's massive amounts of data. And computation all over the place, too. That said, there are plenty of sources of data that you can also use for this optimization process, some of which are still not fully leveraged despite being much older than genomes. There's all the medical data sitting in health records. I would argue we're still not fully optimizing just that data. And then, of course, there's all the data sitting outside of the patient, and the 300 some data sources inside Translator. All of that has to be considered in the context of every patient, too. And so you get this really complex, constrained optimization problem in a very idealized sense, I realize. And what a modern clinician is faced with is this overwhelming challenge. Like they have to somehow solve this problem. They have to choose the right combination of things like treatment and lifestyle to find the optimal health for their patient. That's hard. That's really hard. And that's where all of this is headed when it comes to precision medicine. So to give them some help, you can start to break it apart into different processes or phases within precision medicine. And broadly speaking, there are kind of two phases to it in practice. There's the precision diagnostics phase where you're trying to figure out what is sort of the root cause of a patient's problem. And I've got a genome representing that here because a lot of the work that I do is in rare genetic diseases. So there's usually some genetic variant or genotype that's sitting there at the core. And then there's the precision therapeutics phase. And for the purposes of this talk, I really want to focus on the therapeutic side. It's the side where there's probably the highest unmet medical need. I mean, there's been fantastic advances in precision diagnostics over the last decade. There haven't been equivalent advances in precision therapeutics, and that's really what I want to focus on today. So at a very, very high level, and we're going to come back to this in more detail, there's kind of two ways to think about doing precision therapeutics. There's what I call rational approaches, which will be very comfortable for people with a computational background. And then there's the empirical approaches, where, if you're not comfortable with pipettes, they're a bit scary because it involves going to a bench and doing things with animals. So in the rational approach, you're basically saying, well, what is the core mechanism of harm? What is really driving the disease here? And there's really kind of four directions things go. Like you could have something that's become overactive, in which case you want to inhibit the mechanism. It could be underactive, and now you want to activate it. Maybe it's missing or absent. Now you have to compensate for it or replace it. Or in some cases, something that was fine is now toxic, and so you have to eliminate some kind of product. It would be a biological product or metabolite or something like that. This contrasts to the empirical approach, where you've identified the root molecular cause. And instead of trying to think your way out of it and identify a rational way to invert this mechanism, you just say, you know what, let's just build a model of the disease, probably using genetic engineering or some other technique, and then let's just try things out and see what happens. Both are perfectly reasonable ways to go about this process. Both of them can work in practice. And we do both of them at the Precision Medicine Institute at UAB. Since you're mostly computationalists, I'll show you what a drug screen looks like at a very high level, because we're not really going to come back to it. In a drug screen, you start with the patient once again, and from the patient, you extract a disease model. And it could be their cells that you're using. It doesn't have to be. It could be a cell line you've created to mimic the disease in their cells. That's actually used probably more often. Or you can use an animal model. So it's possible using technology like CRISPR to edit the genome of an organism to match the disease that a human patient has. So CRISPR, if you're not familiar with it, it's like Emacs for a genome. Like you just go and like edit the DNA now. Show me what this does. You compile it. Exactly. It's but with biology. And so you can make a worm that matches the disease a human patient has, or a fly, or a fish, or a mouse. But then what you got to do is you got to study the model and figure out, okay, how does this model reflect that disease? What is its phenotype? And once you have that phenotype, now you have something you can measure the effectiveness of a treatment against. So you can start combining this model with different drugs to see if it actually corrects or rescues the phenotype you observed in that system. Once you have this capability, if it's not too onerous to run these tests, and sometimes it is, by the way, then you just try everything. There's only around 1,500 FDA-approved drugs, and if you expand it to like lots of natural products, we're still only at like 2,600 compounds. It's not a lot in the grand scheme of things. It's what we would call grad student scale. You can have a grad student just pipette 2,600 times over to figure out if something works. And yeah, you're so glad you're in PL now, aren't you? You're so glad. You have no idea what we make students do on the other side. And hopefully, if you just try enough stuff, something works. And then hopefully that thing will actually also work in a patient. And where a lot of the stuff will break down is like you'll get a hit and it rescues the model, but then you'll check the concentration and it'll be like 10 times what a human being can tolerate or something like that. So it's back to the drawing board, or you have to tweak the molecule in some way or do medicinal chemistry. I just want to show that this is a part of the algorithm. So this, an algorithm for me is a step-by-step process that you can follow. In this case, sometimes a step is go to a bench and do something. That's going to happen. So what does this look like in practice when you do it? So now I'm going to show you what we do for a lot of the work at the Precision Medicine Institute at UAB. And our broader mission is what we call science in the service of patients. So the patients that reach out to us are those that, it's a rare genetic disease. They could, in some cases, be the only known patient in the world, or they have a more common condition, may not be that common, but for whatever reason, they've run out of standard of care. It just hasn't responded to the treatments we know about. So these are the kinds of cases that we take on. And really the only criteria we have for getting in is you have to say, look, we've tried everything. And really have tried everything, and then we'll get interested. I've got to give credit to the medical directors here, or the medical leadership team. So we have a lot of medical oversight, on a program like this. We're not just taking results from programs and slinging them back at people or patients. Everything goes through our medical directors to their physician back to patients. So there's a very tightly controlled process in place for all of this. And a lot of the heavy lifting, by the way, is done by undergraduate students. And now we have a program that pulls in medical students for the summer so that they can actually get to work on cases. And really what these folks are doing is they're serving as routers between computational tools, scientists, clinicians, and even the patients themselves, trying to come up with, okay, what's going on here? And based on the tools and experiments and knowledge that's out there, what could be a set of treatments for that patient? They'll write the first draft of these research reports that will eventually go back. Those will get checked by the medical directors before they go out, but it would be impossible to do what we do without their effort. And my hope is that language models may serve as a major accelerant for what these analysts do going forward. So I'll show you some example stories. The first here is from a rare genetic disease caused by mutations in a gene called MAPK8IP3. This patient came to us initially as a 5-year-old boy through the Undiagnosed Diseases Network, through the Duke site in particular. And at 5 years old, this child could not walk or stand, which is pretty typical for this there's like not many patients, so when I say typical, I'm saying typical of the handful of patients known to have this disease. Fairly typical to have pretty severe developmental delay around that age. And so he can't walk or stand, and upon about a year's worth of investigation, they determined that the root cause was in fact that his MAPK8IP3 gene was underactive. So it needed its activity level raised. So the question was, what activates MAPK8IP3? And of course, to answer this question, all you have to do is summon the biomedical data oracle. And so we'll come back to the oracle in a second. And so we summoned the oracle, and the oracle said, oh, just use high-dose retinoids. This will obviously upregulate MAPK-IP3 because look at this research paper over here. There's clearly a connection for how to do this. So we put all this together, packaged it up, sent it off to Duke. They said, yeah, this looks reasonable. We're going to contact the treating physician. And then initially nothing happened, which often happens when you're doing this kind of work because sometimes you're dealing with very complex developmental disorders and it can take time to see something. But out of the blue, 6 months later, we got an email from the mom. She said, you're not going to believe this, but he's actually standing. And not just standing, he's taking some very basic steps now. And again, this is a child that was essentially trapped on the ground or in a wheelchair just a few months before this. So he's about 5.5, I think, at this point. And then a year later, we got another email out of the blue from mom. And she says, you're not going to believe it, but not only is he walking under his own power, but look what he's doing in his physical therapy. So he's significantly better than a year prior, so walking under his own power. And when he falls down now, he can push himself back up. And in just a second, you'll see, I think he goes all the way down to the ground and lifts himself all the way back up. And really, the only thing that changed here was the high-dose retinoids. That was the intervention in this particular case. So that's it's an N of 1. Was it the retinoids that did it? Well, you can't do statistically significant experiments from an N of 1, but nothing else changed. And by the way, we've done lots of follow-on, basic biology that says, yeah, it probably was that. So let's go back, because I invoked an oracle in the middle of this, and it's important to understand what that was. And no, it's not ChatGPT. It didn't exist at the time we worked on this, although I will say that language models do have a role to play here. And as Chris was saying earlier, they are kind of a declarative programming language, if you want to think of them that way. You give them very high-level specifications, say this is what I want, and they're often good at coming back. In fact, what's interesting is this case is not that old. The answer has been known for a while. The information to synthesize the answer has been out there for even longer. But even today, I asked it this morning, if you look for upregulators of MAPK-A3 or activators of its corresponding protein, still can't find them, which to me is a little surprising. But when you get to the long tail of knowledge, sometimes stuff drops off in the language model. So it does happen. So the real oracle in this case, of course, is the biomedical data translator. That's how this was done. I'll add another plug to the interface. Please go test it out. Give us some feedback. And in the context of the cases I'm talking about right now, a lot of this preceded the existence of this interface. I'm talking about stuff we were able to do with our ARA, which is Medicanrin. So I have a lot of thanks to some of the folks in this room for making this particular ARA come into existence and allow us to start tackling these cases early on through the interface that folks like Will were able to build. In a nutshell, you've seen a lot about Translator. The way I put it is it's what you get when you fuse logical reasoning with all the biomedical knowledge you can get your hands on. That's what a system like Medicanrin and Translator more generally is really trying to do, I think. And we can kind of zoom into Translator's brain. And we're going to see obvious places where declarative programming come into play right here. And then I want to, by the end, zoom all the way out to the top, to this sort of the patient-to-pill process as a whole and see, does it apply everywhere? So if you look inside Translator's brain, it's a knowledge graph. So you have all these biomedical concepts represented by nodes. And these are things like genes, drugs, symptoms, phenotypes, diseases. And then you've got relations encoded as edges between them. Okay, that's a knowledge graph. And as an example, there's an edge in there from a paper I published a while back that says Prevacid inhibits NGAs. And you just represent that by tying Prevacid to NGAs with this edge labeled inhibits. So the brain looks like this soup of interconnected concepts, each representing some kind of fact. So it's pretty naturally declarative. To represent a knowledge. It's very natural to represent a knowledge graph in a declarative way. You can come up with these list of core predicates. You can tie things together that way. So you can have relations like inhibits and manifests and decreases and increases and treats. Very, very natural to do that. Once you have that, it's natural to start stitching them together in a very declarative way. So you can say, I could create a may treat relation that says a drug may treat a disease if that gene causes the disease, and in particular, that disease decreases the activity of that gene, and this drug happens to increase the activity of that gene. This is literally encoding the logic we just used for the MAPK8IP3 case. And this template applies over and over and over again for lots of diseases. And what's cool is, you can represent them as things like inference rules, which hopefully provides a launching point for folks in this room. I'm trying to, as I do this, actually try to express everything in something that looks like an inference rule. So maybe we can get proof trees at the end of the day out of everything. And again, because we're trying to convince people at the end of this process, if we can build proof trees, we can always render an argument for a human being to consume. And as another example of a kind of inference you can do once you have a knowledge graph, let's suppose you have two concepts, blue and red. If you know that blue increases red and that red decreases green, then you can infer, logically, that blue should decrease green because you're increasing a decreaser. Once again, you can represent this as an inference rule. And what's great about this is as you add these inference rules, or even more complex rules into the system, you can start to ask really interesting questions over the knowledge graph. I mean, we've seen multiple talks today that point to the potential of the kinds of questions that a system like Translator can answer. So you can ask it, what are ways I might treat this disease? Or you can go very low level, which we often do, and say, I just need to throttle this one mechanism, gene, pathway. Just tell me how to move it up or down. And as another example of that, we have ADNP-driven autism. So this is, it's the autism itself is, it's actually splintering into almost 1,000 odd different disorders, each of which has a different molecular story that links together with a common phenotype at the end. So this is one version of autism driven by mutation in a gene called ADNP. What happened here was early on, like this is early days in Translator, this mom reached out and she said, hey, my son, I knew her before this, but she says, hey, my son's got this ADNP-driven autism. How do you, and we knew by then it was regulated by or caused by haploinsufficiency, so underactivity in ADNP. And she said, how do you upregulate it? Or we inferred that's the question she was trying to ask. And Will ran the query and came back. He just sent me an email almost immediately, said, it just came back with low-dose ketamine. And there's a paper in the cancer literature that it found that connects these things together. So we funneled that back to the chief scientific officer of the foundation, and he emailed back pretty fast and said, gosh, I'd never seen this before, but it looks really compelling. Like the logic here just makes sense that this would actually work. And so by February 2020, they actually had a clinical trial launched at Mount Sinai for this. You may recall something else was happening in February 2020. So COVID blew up, but these parents kept going. And they actually, in the middle of a pandemic, launched a clinical trial. So in September 2020, they actually pulled this off. And a year later had completed it. And this is the crazy part. The core prediction from Translator was validated. We did see ADNP levels actually normalize under low-dose ketamine. But more importantly, the stuff that actually matters, like the patients actually got much, much better. So I think there's a huge number of endpoints measured here, and all but one, the one being, I think, insomnia, improved in a statistically significant fashion. If you want to know more about the patient side of this, there's a great story in Stat that covered all of this. And then a year later, we finally caught up with, the publishing side of it. So this is frequently the case in precision medicine. Like the time from prediction to actual publication, I think it was like 3 years. But the time from prediction to helping patients was far less. And this happens over and over again. So another case, this one you might have actually seen on Netflix. So there's a series called Diagnosis. And in this particular case, she was the first patient in the world ever diagnosed with this disease. She was suffering from somewhere on the order of 150 cataplexic episodes per day. Cataplexic episodes look a lot like seizures. They're not seizures. There's a different underlying mechanism. But you can think of them as she just kind of froze up repeatedly over the course of the day. And kind of scary was she would stop breathing during them too. So these are pretty dangerous episodes. And so, of course, we said, well, let's check Translator. Let's see what it says about this. And in the humorless way that Translator sometimes often does, it just says, just give her crystal meth. It's fine. She will feel so much better. And also, maybe some spider venoms and plant toxins. Well, because sure enough, these are actually inhibitors of the overactive target. It's not wrong. It might work in cells. And the question is, does it work in people? So in the time where we're trying to figure this out, and actually we were doing empirical drug screens, 3 more patients popped up. And as I talked to one of the dads on the phone, he's like, oh yeah, by the way, like my son is like so much better, and it's because he takes this ADHD drug now. And I went, oh no. Let me guess which one it is. He goes, yeah, it's one of the amphetamines. So sure enough, crystal meth wasn't. It's right. It is actually an inhibitor of the overactive channel in this case. Which is KCNMA1, and this is the ADHD drug, and this is what translator had recommended. So it differs by a methyl group, and that's not from like a medicinal chemistry point of view. That wasn't the important part of the molecule. That was not what made the difference. It was actually the amphetamine part of the amphetamine that actually made the difference. And so now you have a lot of drugs to choose from. So it turns out all the amphetamines work for these patients. So we sent a report over to the physicians, and the mom called like the next day, and she said, by the way, it worked. And because you get an instant feedback on this one, like within like minutes, it's working. And from 150 cataplexic episodes to 0 episodes. So extraordinary effect size as well. And now we know of at least 5 patients where we see exactly the same effect. And so we are actively working with the clinical center at NIH and trying to find some funding to do a very unusual clinical trial, where what we would do is, take these patients to the clinical center, give them a short-acting amphetamine at the start of the day instead of a long-acting one, which is what they take, and then look at cataplexic episodes versus blood levels of amphetamine over the course of a single day. So we think we can get enough confidence from that to say, look, this is what's actually causing the reduction in episodes. Another example is COVID. So we did a lot of work here. So as soon as COVID hit, we started working with all the data coming downstream from all the research across the world, just pumping data out on COVID. We started distilling as much as we could into drug targets to go after, because this is a great, translator is just a beast when it comes to chewing apart drug targets. We can start feeding them in and saying, give us drug recommendations plus evidence that this is a good thing to do for COVID, and then farming those out downstream to either testing in cells, testing in clinical trials, or straight into animals. Within, I think by March, we had 40 predictions for things that could work. By early April, we had 20 tested in an early cellular model, and a high priority one emerged right away, which was anti-androgen therapy. So we took this to the VA, and by April 2020, they said, yes, we're going to do a clinical trial. What we're going to do is chemically castrate veterans that have severe COVID, and because that's what the anti-androgen therapy is. For some reason, not enough veterans wanted to do this. They said, they're on their deathbed. We go, we can chemically castrate you, and it might save you. And they go, I'll take my chances. So the crazy thing is, the idea did work, though. It just took longer to figure this out. And what happened was other groups used much milder anti-androgens, like hair loss drugs. They should have started with the hair loss drugs. That would have been a much easier sell. And those do work. So there's plenty of evidence now that anti-androgen therapy does work for severe COVID. So by the way, we're on the record, at least on Twitter or now X or whatever it's called now. Like you could look up the tweets where we were the first ones to claim that this would work. So we should get credit for that. And the last case I'll show you today is actually my own personal case, or really my son's case. And this is why I'm not in PL anymore. It's why I haven't been here in 6 years, guys. It's because I was trying to help my son. So by the time Bertrand was 6 months old, I knew something was wrong. And our family had landed on a place that you might call undiagnosed island. Actually, you'll hear this term a lot in the rare disease community, where people are in the undiagnosed phase, and they're not really sure what's happening. And while Bertrand was there, he had seizures, he had a movement disorder, severe developmental delay, and for some reason, a lack of tears. And I'll be honest, from a day-to-day basis, one of the most challenging symptoms to deal with was the fact that he wasn't making tears. Because if you don't make tears, your eyelids will abrade your cornea. Your eyes will get infected. And it was to the point where he was having eye surgery to drain pus out of his eyes from the lack of moisture. By the time he was 1, he had a life expectancy of just 2 to 3 years, based on the constellation of symptoms that he was facing. And you just got lucky that around that time, a breakthrough occurred in genomic science. Something called exome sequencing became possible. This is where you can economically look at the fraction of, like, it's about the 1 to 2% of a human genome that encodes proteins, where we think 80 to 90% of all genetic diseases actually live. And so it was suddenly economical, for some definition of economical, to look at this and try to figure out which mutations could actually be driving a disease like this. And through this study, it was actually conducted at Duke, they identified 2 mutations in a gene called NGLI1. And after this, Bertrand became the first NGLI1 deficiency patient ever discovered, which by extension means he was the only one at the time that was out there. And I knew to do anything about this, I was going to have to find some others. Fortunately, as a computer scientist, I had access to a secret weapon, Google. Now, I obviously couldn't just Google for more patients. It's not quite as simple as just checking to see if there's others that are out there, because if you're the first in the world, there aren't any. So it's not that simple. What I did instead was search engine optimization using a blog post. And here, all I had to do was just get a blog post to rank highly in search results and to go viral so that people would look at it and check to see if it looked like something that they knew for their child. So this is the top part of that blog post, by the way. So the link bait title, the picture of Liam Neeson, like all that's there to try to get it to go viral. But there's like 3,000 words down here that was all about search engine optimization. And it actually worked. So for people that didn't have tears, this is one of the places they find. So I'm like an expert in telling people, no, you don't have NGLI1 deficiency, you actually have Sjogren's, which is the thing you usually have when you don't have tears, because people reach out for that all the time to this day. But sometimes they are NGLI1 patients. And in this case, it took two weeks to find 2 additional patients. They happened to be over in Turkey, a sibling pair. For me, it led to a jump into glycobiology and into unusual collaborations I never would have imagined, like with this guy here, Hud Freeze, because the disorder itself was what we call glycobiological in nature. So I had to learn a lot about this. I was learning a lot of genetics, now I had to learn a lot of glycobiology, and this guy helped me out. If you want to know more about this phase, there's an article in the New Yorker that covers a lot of it and talks about the emergence of this disorder, the discovery of the community, and all of that. Got me involved in the Undiagnosed Diseases Network. I'm now one of the PIs on the coordinating center for this many years later. It led to NIH launching a natural history study and in pulling these kids into the clinical center 1 week every year, collecting massive amounts of data on these patients and leading to very useful insights. I ended up getting, I was at Utah at the time, ended up picking up a secondary professorship in pharmaceutical chemistry. And I'd learned just enough after about 2 years of this to make a prediction. And it's something I think we could really automate at scale now. And the prediction was, okay, if NGLI1 breaks, something else has got to break too. That was really the logic I was going after. And what I was able to figure out is that from this, Bertrand is missing something. He's not just missing NGLI1. NGLI1 has a function. It turns this thing into that thing. And so if the producer of these things goes away, you have a deficiency in something. You also might have an accumulation of something. That's the fundamental logic you have in what we call metabolic disorders. So the thing in this case was N-acetylglucosamine, which at the time meant nothing to me, but it was a series of characters that I could type into Google. And so I did that and realized that you can actually buy N-acetylglucosamine on Amazon. So I did that. I got like a bag of it 2 days later at my doorstep, and decided that the next step was safety testing. So I just literally ate the bag myself, and woke up the next morning alive. And at that point, I was like, well, I guess FDA phase 1 safety testing is complete. Now, I'll mention now, It does cause horrific osmotic diarrhea at those doses. But I was alive. And I still had sort of that like Star Trek level ethical dilemma. That I was like, do I give this to Bertrand? I'm not really sure. But he made it simpler because he went to the hospital for pneumonia on something unrelated. Came very close to dying, which he would do frequently. And I realized, like, man, if I'd had this sitting on my shelf and I never found out what it might have done for him, I wouldn't be able to live with myself as a parent. And so once he got home, once he was better, I said it's time to try it. And I didn't know what I'd see. I didn't know if it would help with seizures or development or movement disorder. But in the end, what it helped with was tears. So about 3 days into trying this out, I was giving him breakfast one morning. I was running behind. And so when I got up to his room, he was crying, but he was actually making liquid tears, which up to that point in his life, I'd never even seen. So I did what I think any parent does when they see their child cry for the first time. I collected his tears, I packed them on dry ice, and I shipped them to a lab in California for analysis. So Hud Freeze did proteomics or glycoproteomics on Bertrand's first tears. And those tears really ended up being kind of an ocean of science for the disorder as a whole. We learned a lot about this disease from those tears. And I learned quickly that biologists are very strange, because most people are like, hey, that's great, that you can help your child. But biologists say that's great, but they say we are biologists, and the important question is, will it work in flies? And well, you can find out. So I partnered with Clement Chow, who was on the previous slide. We built a fly model of the disease using genomic engineering. And sure enough, if you give them this disease, the flies don't do very well. About 18% will survive. But if you start giving them this compound, and you keep giving them this compound, then around 80 to 90% will survive. I don't know if the flies cry. I'm not even sure if flies can make tears. But they do survive, and that's probably a lot more important in the end. If you want to know more about this, all this work is now published too. This led to some other rapid changes in my life. I ended up getting invited to sit on the faculty at Harvard Med School in biomedical informatics. Still there as a senior lecturer to this day. And I co-founded a company to start doing personalized epilepsy treatments. And this one was empirical. We just literally built models of patients, cellular models, and just tested everything. That's all we did. And then measured it using patch clamp electrophysiology. That actually worked really well. It was a dumb idea that actually worked really well for patients. So that company got acquired in 2018. And the company that acquired that company got acquired later on, just back in January. And then I got the strangest offer ever, which was to help launch the Precision Medicine Initiative. So I ended up getting invited to the White House, and Obama says, hey, I'm going to launch this Precision Medicine Initiative. Do you want to help architect this thing? And I didn't say at that time that I'm actually an expert in static analysis and you should talk to somebody else. I just said, sure, why not? Might as well. And that led to me helping to craft some of the founding white papers by the launch of the initiative. I ended up working at the White House for 3 years. Today, this program is still around. So if you've seen this All of Us program, that's what the Precision Medicine Initiative became. It's collecting 1 million genomes and corresponding health records so that we can build what I would think of as a Rosetta Stone for the human genome. I helped out a lot on the Million Veterans Program, a sort of sister initiative to this that started actually a little bit earlier. Helped out in Biden's cancer moonshot, did some work on helping to pass 21st Century Cures, which ended up funding the Precision Medicine Initiative. I got to launch a little micro project on precision medicine for actual patients inside of the Precision Medicine Initiative too. For reasons I still don't quite understand, I was asked to stay on for a year after the election. So I did. And that was an interesting year. But after a year of that, I went ahead and quit. And sadly, I ended up quitting PL2. And not by choice, but just because the scale of the opportunity to make a difference in human lives was suddenly greater. And that's because I got an offer to come create this Precision Medicine Institute at UAB. And it was a ridiculous offer in terms of the resources they were putting on the table. And it was a chance to immediately try to replicate the kinds of things I was doing for Bertrand, but for lots of others. And the reason I could even show you those stories I showed you, that's all because they ponied up and said, let's do it right now. They made that possible. Yeah, of course, I kept helping Bertrand, and he continued to define, what eventually I would start to call the algorithm for precision medicine. So I got small grants to actually do real drug screens for NGLI1 deficiency, and to do this, we actually made NGLI1 worms. And what we did with these worms, they're planarian worms. They're great models of developmental disorders, by the way. I won't go into the biological details. That's not really relevant for this audience. The important thing is they showed us another drug target, something else we could go after. And this is an important part of the algorithm itself, is that sometimes you start off where this is the core mechanism of harm, but then when you really investigate it, you say, well, there's something else downstream that we can go after instead. And that's much easier to hit. And that was absolutely true in this case. The downstream target in this case was NGAze. And this allowed us to do some more computational work, a kind of molecular simulation called a docking simulation. And my shorthand way of describing this is you take a 3D model of a protein, your drug target, and then you shut off regular physics and you import video game physics. So literally you do, and then you get to use BSP trees and octrees and stuff like this that makes certain things in video games really, really fast, whereas simulating real physics would be really, really slow. And you do the same thing. So you treat the protein like a rigid structure, and you look for molecules that are inverse in shape and charge to the catalytic domain or the active site on whatever this particular protein happens to be. And then hopefully that's faithful enough to what happens in reality. You can think of it like an abstract interpretation of physics, I guess. And that might give you a sense of what actually works. So you're trying to dock things with some now rigid region on this protein. In real life, proteins, they bend, they wiggle, they wobble, they can change conformations, they can do things like that. But for this stuff, you just say, don't let them do that. So we screened around 200,000 compounds this way. 70 looked like they actually work, or would actually potentially dock. 14 of them were already FDA approved, and one even worked in the lab. And that happened to be Prevacid. So you can buy Prevacid at Costco. I did. If you want to know more about the science and all this, by the way, that's all written up too on everything from doing the docking simulations to testing it, the worms, all that is in here. There was a company at the time, Perlara, that had built a different fly model for NGLI1. They said, hey, we got a fly model. We'll just throw it on there and see what happens. And Prevacid just so happened to double survival in their particular fly model. Of course, we've run, NGLI1 through Medicanron, and interestingly enough, when we ran some of the secondary targets that had emerged through the basic biology one of the top hit was actually broccoli. Not broccoli itself, but like a component within broccoli called sulforaphane. And then this is, again, remember at the start I said sometimes you need doses higher than you can achieve? Well, this was the case here, because you need like a dump truck worth of broccoli each day to pull this off. Luckily, you can get just the sulforaphane condensed down into individual pills, so you can actually do this. And over time, these sorts of interventions started to really stack up and make a difference in Bertrand's quality of life. So if you want to measure success, the metric I like to use in precision medicine is the one you measure in smiles per hour. Like, what are we doing in terms of making a difference in the overall happiness and well-being of our patients? And that's a much more realistic metric to use, and also a practical one. Not every patient we work with is going to suddenly be normal overnight, for whatever normal means anyway. But we can often make a big difference in their day-to-day life. That we can do. In Bertrand's case, it meant things like qualifying to use an eye gaze assistive communication device, which he, up until this point in his life, had never really qualified to use. And what it enabled him to do was really express himself for the first time. So he was able to say things like, when I would come home, hello, like Matt is my dad. And this was Bertrand's language for saying, hey, welcome home, Dad. So we'll come back to Bertrand in a moment. I want to circle back to the algorithm itself, because we've seen lots of individual examples at this point. And lots of ways to go from a patient or data over to a target, over to a drug. And what I want to know now is there a general process, a repeatable process that allows us to do this over and over again? What I'm asking in some sense is there an algorithm, a step-by-step process that we can follow? And so I used to give talks for years where I would show something that looked kind of like an algorithm in high speed. I would just kind of wave my hands, and so I'm going to do it again. Here's something that looks like an algorithm, and I'm waving my hands, and it's all moving by in very high speed. And it's a flow chart, and it's not wrong. It's just that this formulation of it misses things. And so what this high-speed algorithm, what this flow chart is doing is saying, try to figure out the impact. Is it gain of function? Is it loss of function? Is it absence of function? Is it toxic? And then it goes into all the different things you could do down each of those paths. And that was an early simplistic encapsulation of it. And then I realized as I was writing it out that it doesn't capture everything. And if you look at what happens in this algorithm for precision medicine, there's some key features here. So first of all, it's not deterministic. So there can definitely be multiple reasonable next steps for any patient. There often are. You don't have to do just this thing or just that thing. For example, take the high level. You don't have to do just the rational thing or just the empirical thing. You can do both at the same time. So the algorithm is implicitly parallel. And for example, if you had a patient with a total loss of function in some gene, it's perfectly reasonable. Often the case that we'll say, the two things you want to do right away is do some RNA sequencing on iPSCs or derived differentiated cell cultures, and also at the same time, build a model organism, because either one of those might be crucial down the road for what you're doing. It's recursive. There's lots of ways that the algorithm can interleave with itself. So for example, if you go back to the high level and you look at the rational approach where I'm saying, study the mechanism and try to figure out which direction to go, you might end up doing some science where you actually discover there's a downstream mechanism, and now the whole process repeats, but for the downstream mechanism. And by the way, the process by which you discover the downstream mechanism, maybe that was empirical, but it also could have been strictly bioinformatic. You might have done queries on some pathway database and realized that, hey, there was something downstream. Or you look at gene regulatory networks. There's lots of ways you can end up stumbling across downstream mechanisms of some root cause of harm. And then, of course, sometimes parts of the algorithm are empirical. Like you actually have to go to a lab and do something. And so we need some framework that can concisely express all of this. And by the way, by empirical, I mean something like, build a zebrafish model using CRISPR, Emacs its genome, and then throw drugs on it and see what happens. That's a thing that happens all the time. So there is an English language description of all this. Like if you go to bertrand.mite.net, you'll find an 86-page description I've written of what this process looks like. It's something that I frequently reference when patients reach out to me and say, here's some light evening reading. Go look at this and let's try to figure out where you are in this process, or all the places you could be in this process. And the thing is, it's not very imperative. I tried actually. I was given a talk a few months ago. I was like, I want to see if I can write down like an algol-like pseudocode version of this, and it got really painful. I was like, I'm putting like fork in here, and I was like, adding this imperative or this thing where I'm like, okay, now you've just learned this fact and maybe trigger other actions based on learning this fact. And it got so awkward, I just gave up on it. I said, it's not the right way to express this process. So I think it's better to think declaratively. And so let's just start all the way at the beginning. What are we really trying to accomplish? We want to know the answer to this question. For all the X's that are out there, could X be a treatment for some patient? And I think the simplest possible way we could try to expand this definition in a declarative way would be, X may treat this patient if this patient has a specific disease, and it's known already that this drug treats this disease. And this sounds probably silly, like why would this even be necessary? Except that sometimes even something this simple actually works very well. And so I'll tell you Kelsey's story as an example of this. When she was 19, her parents reached out because she was suffering from severe cyclic vomiting syndrome. She weighed 75 pounds and was suffering dozens of vomiting episodes every day. Of course, her physician said, we've tried everything, which we often hear. In their case, when they said we've tried everything, What they really meant was about 25 things. They had tried 25 things for Kelsey. So we checked everything. Of course, we have access to Translator. We can look at hundreds of different data sources to see what could be out there. This includes like 30 million published papers, of which there's 64,000 that talk about nausea, and 4,000 of them that talk about treating nausea. And between them, you can find 347 distinct proposed treatment modalities for nausea. So here's what everything actually looks like when you look at everything. What you get is this long tail, and there's regions inside of it. So up at the top, you get Zofran as the most popular, most cited treatment for nausea anyway. But if you zoom all the way down here, you will find in the long tail really unusual stuff, like nasally inhaled isopropyl alcohol as a treatment for nausea. And this actually, as it turns out, was what we needed for this case. So if you zoom back out, because like their physicians, her physicians team, they hadn't heard of this potential treatment. They knew about all the stuff that was at the sort of the fat part of the long tail. They knew about that because that's what most physicians would learn in the course of medical school. And then there's everything that humanity actually still knows, the unknown known, but which any individual physician might have forgotten. So there's the known known, the stuff we know about, and the unknown known, the stuff that is known to our species but lost to any one person. So we sent those papers over, and her physicians were convinced to try it. And we got feedback the same day that it seemed to be working. Suddenly she was able to arrest every episode of nausea from there forward. And if you jump forward 8 months, not only was she still no longer vomiting, being able to intervene like this all the time, but she was up to 125 pounds and got married. And it's because she was able to get out of the hospital. Her high school sweetheart proposed to her, and they even moved to California, where she's living very happily now. So again, even though this seems really simple, it's a good place to start because it can actually work. And we can sort of drill down from here into other forms that get more specific. So for example, if we're dealing with rare genetic disease, we might expand it a little bit. We could say, X may treat this particular patient if they have a particular pathogenic genotype, and it's known that this genotype will push up activity in some gene, and this drug is known to push down activity in this particular gene. So then we can sort of break it down. Are we out of time? Okay. So we'll just stop here. Because I'm basically, all I was going to do now was just show you more and more examples of how you can phrase fragments of this in a declarative way. So how you can think about defining inhibits, how you can think about defining enhances, and I can share the slides. So if you have to go out and do something in a lab, it's in blue, and if you have to just look it up in a database, it's in green. And I was able to define, as I went, more and more of these 86 pages in this style. So I will stop there and take any questions if we have time.